Happy studying, friends, and welcome back to Easy Dentistry. We have started uploading lectures on microbiology as this subject was highly requested by all of you. In this lecture, we will be studying the basic concepts of immunity part 2, that is the acquired immunity. In our previous video, we have discussed about the innate immunity. Hope you all have watched that video. If not, I highly recommend you to watch it before proceeding with this video. The link of innate immunity I am giving in the description box below, so go and watch it. So let's begin, but before that, please subscribe to our channel and also hit on the bell icon. Do comment and let us know your feedback, and don't forget to share these videos with all your friends. So, beginning with acquired immunity. By definition, acquired immunity, as the name suggests, it is the resistance which is acquired by an individual during his life. So, the acquired immunity is of two types, the active and the passive acquired immunity. Now, talking about the active acquired immunity, the active acquired immunity is again of two types, the natural active acquired immunity and the artificial active acquired immunity. The natural active acquired immunity occurs due to subclinical or clinical infection in a patient, while the artificial acquired immunity is introduced through vaccination. Now let us have an overview of the acquired immunity. When this individual comes in contact with an antigen, this antigen may be in the form of any natural infection or in the form of any vaccine. Then there is stimulation of the immune apparatus in this individual, and there is formation of either antibodies in the individual's body against these antigens, or there may be activation of the immune cells in the body against these antigens. So, the active immunity develops after a latent period. Now, what is this latent period? It is the time needed for the immune system to develop. Now, once this active immunity has been developed after the latent period, it is long lasting. Now, the mechanism of active immunity. The active immune response stimulates both the humoral as well as the cell-mediated immunity, and usually this stimulation occurs parallelly, that is, both the humoral or the antibodies and the cell-mediated immunity or the immune cells are activated simultaneously. Now, what is humoral immunity? Humoral immunity is the antibody-mediated immunity. It depends on the synthesis of antibodies by the plasma cells. These plasma cells, they produce specific antibodies, and these antibodies, it combines with the antigens, and this modifies the activity of the antigens by causing either the lysis of the antigen or the neutralization of the toxins released by the antigen or the phagocytosis of the antigen. Now, the cell-mediated immunity. The cell-mediated immunity, it depends upon the T lymphocyte activation by the antigens. The antigens, when they enter into the body, they activate the T lymphocytes, and these T lymphocytes are responsible for the cell-mediated immunity. Now, this cell-mediated immunity play a major role in the chronic bacterial infections. So, in chronic bacterial infections like tuberculosis, leprosy and viral infections, the organisms, they multiply and survive in the phagolysosomes, that is this is called as intracellular parasitism. So, this cell-mediated immunity is very much active, and it is very much important in the chronic infections like the tuberculosis, leprosy and viral infections in which there is intracellular parasitism. Now, the types of active immunity. active immunity is natural active immunity and artificial active immunity. That this is two types of active immunity. Now, the natural active immunity, it is acquired by a natural subclinical or a clinical infection. So when there is a natural clinical or subclinical infection in an individual, he develops natural active immunity, and this immunity is very much long lasting. Example of this is the persons recovering from smallpox infection develop natural active immunity. Then is the artificial active immunity. The artificial active immunity is produced by vaccination. Now, these vaccines are prepared from either the live organisms or the killed microorganisms or the toxoids of these microorganisms. Now, how are the organisms killed? The microorganisms are killed by either heat application or use of formalin, phenol or alcohol. Now, these killed organisms are preserved either in phenol or N-merthiolate or the alcohol. Now, the toxoids of the microorganisms are prepared from the bacterial exotoxins inactivated by either formalin or alum. The toxoids prepared by inactivation with the use of formalin is called as formal toxoids, while when the alum is used, it is called as alum toxoid. Now, the vaccines are of three types: the live vaccine, killed vaccine, and the bacterial products. The live vaccine examples are BCG, which is used for tuberculosis, TY21A used for typhoid, Sabin vaccine, which is used for poliomyelitis, MMR used for measles, mumps and rubella, and 17D, which is used for yellow fever. So you should remember the names of these vaccines, live vaccines, because it is mostly asked in your exams. Now, the examples of killed vaccines are TAB, which is used for enteric fever, killed cholera vaccine, Salk vaccine, which is used for poliomyelitis, neural and non-neural vaccines, which is also used for, not polio, but it is used for rabies and the hepatitis B vaccine. So these are the examples of few killed vaccines. So you should remember all these names. Then the bacterial products which are used for vaccination are the tetanus toxoid, which is used for tetanus, and the diphtheria toxoid, which is used for diphtheria. Now, after studying the active acquired immunity, let us study about the passive acquired immunity. Now, this passive acquired immunity is again of two types, the natural and the artificial. The natural passive immunity is through the transplacental maternal immunoglobulin G antibodies, while the artificial passive immunity is acquired through the use of the anti-serum injections. Now, the passive immunity is induced in an individual by injecting the preformed or the ready-made antibodies in this individual. So, how are these ready-made antibodies formed? Another host is injected with an antigen. Now, this host produces antibodies, and these antibodies or anti-sera are then injected into this infected host. So, this infected host receives ready-made antibodies from another host. So there is no active role of the immune system of this individual, and he develops passive immunity against that infection. So this is the mechanism or a gist of what is passive immunity. Now, the immune system, it has no active role in passive immunity. This passive immunity provides immediate protection against any infection, there is no latent period, but this passive immunity is short lasting. Now, this passive immunity is again of two types, the natural and the artificial. The natural passive immunity is acquired through transplacental route or through the colostrum, while the artificial passive immunity is through the parenteral antibody administration. The agents which are used for artificial passive immunity are the hyperimmune sera of either animal or human origin, the convalescent sera, and the pooled human gamma globulin. These are the agents which are used to produce artificial passive immunity. Now, the oldest method to produce this artificial passive immunity is the use of the horse anti-sera. Horse is injected with a series of tetanus toxoids, then bleeding is induced in the horse and the serum is separated, and this serum contains the ATS or the anti-tetanus serum. But the horse ATS, when used in humans, may produce hypersensitivity reactions. So, to avoid these hypersensitivity reactions, human anti-tetanus sera are produced, in which the humans are injected with a series of tetanus toxoids to form human ATS, and the action caused by this human ATS is long lasting and there is no hypersensitivity. Now, what is convalescent sera? It is the sera of patients recovering from the infectious diseases. They contain high level of specific antibody and therefore is employed for passive immunization against measles and rubella. Now, what are the uses of this passive immunity? Another important point you should remember, the uses of passive immunity is to provide immediate short-term protection in non-immune host who has an increased threat of infection. Secondly, it is used to suppress the active immunity, for example, in Rh incompatibility cases in which a Rh negative mother carries an Rh positive fetus. In these cases, the Rh immunoglobulin is given to the mother during delivery. This prevents immune response to Rh factors in the babies. Now, the treatment, it is also passive immunity, passive acquired immunity is also used for the treatment of certain serious infections. Now, let us see what are the difference between the active and passive immunity. This is a very important question which is asked in exam. So, watch this part of the lecture carefully. Now, the active immunity, it is produced actively by the immune system, while the passive immunity is received passively by the individual. The host immune system do not take any active role in producing the passive immunity. Now, the active immunity is induced either by an infection or by contact with immunogen, while the passive immunity is passively conferred by administration of ready-made antibodies. The active immunity is long-lasting and there is effective protection, while the passive immunity is short-lived and less effective. The active immunity is effective only after a lag period, which is the time required for the generation of antibodies, while the passive immunity is effective immediately. Now, the immunological memory is present in active immunity, therefore subsequent challenge is more effective, and the subsequent challenge appears as a booster effect, while the passive immunity has no immunological memory and subsequent administration of antibody is less effective due to immune elimination. Now, in active immunity, the negative phase may occur. This is due to antigen combining with already existing antibody and lowering its effect, while in passive immunity, there is no negative phase. In active immunity, This active immunity is not applicable in immunodeficient persons, while in passive immunity it is applicable to the immunodeficient persons. Active immunity is used for prophylaxis to increase body resistance, example the BCG vaccine, while the passive immunity is used for treatment of acute infections. Now let us study certain miscellaneous terms. What is combined immunization? Combined immunization is a combination of both active and passive immunization which is employed simultaneously. The passive immunity provides the protection necessary till the active immunity becomes effective. Now, what is adaptive immunity? Adaptive immunity is the injection of immunologically competent lymphocytes, and this injection results in the formation of adaptive immunity. It is used in the treatment of lepromatous leprosy. Such lymphocytes are called as transfer factor. These transfer factors is an extract of immunologically competent lymphocytes which is used to acquire adaptive immunity. Now, what is local immunity? Natural infection or live viral vaccine administered either orally or intranasally provides local immunity at the site of entry. Immunoglobulin A plays a major role in this local immunity. Now, herd immunity, it refers to the overall resistance in a community. When the herd immunity is low, there is increased chance of epidemic, while if the herd immunity is high, there is fast eradication of the communicable disease. So here we complete with the basic concepts of immunity, in which we have studied both the innate immunity in our previous lecture and the acquired immunity in this lecture. Watch those lectures carefully. Now it is the MCQ time. If you have watched these two lectures carefully, you will answer with all these MCQs. The first question is, clinical or subclinical infection leads to option A, active natural immunity, option B, passive natural immunity, option C, active artificial immunity, option D, passive artificial immunity. Note down your answers in the comment box below. Second question, vaccination induces A, active natural immunity, B, passive natural immunity, C, active artificial immunity, D, passive artificial immunity. The third question, artificial passive immunity may be induced by A, anti-serum injections, B, vaccination, C, maternal antibodies, and D, all of the above. Fourth question, all of the following are live vaccines except BCG, Sabin vaccine, MMR, and TAB vaccine. Fifth question, all of the following are killed vaccines except Salk vaccine, hepatitis B vaccine, non-neural vaccine for rabies, and the MMR vaccine. Write your answers in the comment box below. The next lecture will be on antigens, which is another important topic. That's all for now. Happy studying.